So we ultimately aim to increase our chances of success in the clinic by precisely identifying the right targets in the right cells for the right patients, whether that be in autoimmunity or oncology. So it's extremely challenging to find good targets for IBD because, like many other diseases, it's biologically complex, with aberrant genetics, the environment, and the immune system all interacting in a way to drive disease pathogenesis. Even with a lot of data, it's difficult to distinguish what is causal and what's associative. Moreover, sparsity in clinical metadata leaves us with underpowered insights. In addition, most model systems don't actually reflect patient-level heterogeneity or complexity. What works in a dish or a mouse might not actually translate to what's happening in a patient. So at Immunai, we believe that our approach really addresses finding the right target for the right cell type and for the right patient. This approach is clinically informed, where we start with data from hundreds of real patients, integrate this data into a high-resolution single-cell atlas, and after this harmonization, we're able to map thousands of cell types to clinical states, which enables statistically powered insights. These insights are driven by our AI models, which are able to cut through the noise to pinpoint genes driving key disease processes like inflammation or therapeutic resistance to mainstay therapies. We then are able to perform disease-relevant validation for compelling candidate targets. We test targets in models that better mimic human disease, and then we can map back the effects to real patient data. I think this tight loop is really key. Going from patient data to functional validation and back is really what is solving a core issue here, where traditional validation does not reflect key patient biology. An example of a target that stood out using this process was a gene that we found linked to inflammation and anti-TNF non-response in IBD, especially in macrophages and CD8 T cells, which are two key immune cell types driving the pathogenesis of disease. When perturbing this gene, it reduced inflammation and other disease-relevant processes, but only in macrophages. And this is really powerful. We were able to determine which cell type was most relevant for this gene. When we mapped back these results to patients, we saw that the gene's perturbation resembled most closely anti-TNF responders and healthy patients. So we ultimately aim to increase our chances of success in the clinic by precisely identifying the right targets in the right cells for the right patients, whether that be in autoimmunity or oncology. By combining real patient data, AI models, and functional validation, we've built a repeatable and adaptable system for target discovery. We can understand core immune mechanisms and drivers of immune dysfunction that then connects to many diseases. And ultimately, each time we do this, we can feed back those insights into the system to make it better. We think this approach is really key to driving the future of drug discovery.